The overall goal of this procedure is to quantify CpG methylation in targeted regions of interest out of genomic DNA isolated from tissue or cells of interest. This is accomplished by first designing bisulfite-specific PCR primers to amplify regions of interest from bisulfite-converted genomic DNA. In the second step, the bisulfite-converted region of interest is amplified and next-generation sequencing, or NGS, libraries are generated from the amplicons. Next, the libraries are sequenced and FASTQ files are generated for analysis. In the final step, the sequencing reads are aligned to the bisulfite-converted reference target sequence and the 5MC% is calculated. Ultimately, bisulfite amplicon sequencing, or BSAS, can be performed to precisely and accurately quantify the CpG methylation in any region of interest from any model organism.
The main advantage of this technique over existing methods, like Sanger sequencing, is that BSAS provides accurate and precise CpG methylation quantitation in a rapid, high-throughput manner, providing the opportunity to assay multiple regions in a high number of samples.
After identifying the target and designing the primer, assemble the reaction for the target amplification optimization of a single amplicon, and then seal the PCR plate with heat-sealed film. To visualize the amplicons by phage electrophoresis, next load the appropriate wells with 30 microliters of PCR reactions or ladder, and then run the gel at 200 volts for approximately 45 minutes. When the first